Just we had an introductory part of the excretory system, and only Nikita, you were present, isn't
हो
it?
सर.
And that too, after some time you left.
हो.
Why? Whether is there was there any problem of network? What was the
सर
problem?
माझा प्रॉब्लेम होता त्याच्यामुळं लेफ्ट म्हणजे खराब झालेला. आता मोबाईल चेंज केलाय मी.
Okay, because I was seeing you, but you are not responding. That's why I'm asking.
हा होतो जरा त्या फोनचा खूप प्रॉब्लेम होत होता त्याच्यामुळे
Okay, okay,
चेंज केला घेतलाय.
okay, okay. So, but let us have a revision what we have discussed in the last lecture. These are the points which we have actually uh, considered while dealing with the last lecture, because we are going to cover two systems here, excretive system and the reproductive system in today's lecture. But out of this, we have already discussed on the excretive system. But even though, let us have a revision, and or we will have an entire lecture again, because let others also can come to know what we have discussed in the last lecture. So excretory organs, there are two main excretory organs. One is kidney and another is the Kebers organ. So we will discuss one by one on its structure and the location. So first of all, kidney. They are also called as the organs of Bojanus because of the name of the scientist who have discovered these kidneys in the body of bivalve. So where the location is, this is the diagram which we have come across while studying the respiratory system. This is the sectional view of the bivalve. This is dorsal side, this is anterior side, and this is right and left side. This is the location of the foot, which divides the entire mantle cavity into two. And in the mantle cavity, major part of the mantle cavity is occupied by the gills or tenidia, which are W-shaped. See, this is the W-shaped gills are present. And we have studied in details the structure of the gills in the last lecture, which is having two gill lamina, outer and inner gill lamina, and these two are respective inner inner outer gill lamellae of the respective gill lamina. Okay. Now the location, see location of kidney. This is a pair of kidney. As the animal is bilateral, lies on the either side. This is in the sectional view. We have what we are observing here in the sectional view. But actually, how the kidney appears if you observe it from the side view, what the structure shows. Each kidney is a long, dark, and the glandular tube. This word tube is very much important. Tube open at both the ends. It is bent upon itself like a road. How the road turns on itself in the ghat section, like that U-shaped tube is there, and the loop at the posterior side, loop the loop at posterior side and both ends at the anterior side. See, this is the location. This is the U-shaped loop. This is the loop at the posterior side, and these are the two ends at the anterior side. That is what this word means. The loop at posterior side and both the ends at the anterior side. The two limbs of the loop lie parallel one above the other like this. This is one limb, this is another limb. They are lying parallel to one another above one another. This is dorsal, this is ventral loop as such. So this ventral loop is known as kidney proper. The two part means kidney proper and the ureter or urinary bladder or ureter. So kidney proper, which is lower, this one. This is actually diagram in the book. This is a diagram sketched. See, this is the lower part, that kidney, and this is the upper part. This is the upper part, that is urinary bladder. This spongy and glandular. It is thick walled, opens anteriorly into fluid filled pericardial cavity. See, this is the pericardium in which the heart is located, which is shown in the red dots. See, this is the auricle, this is the ventricle, which is enclosed in the pericardium. The digestive system, part of the digestive system, intestine passes through the ventricle, which we have already mentioned that it is the typical nature part or the peculiarity of the digestive system passing through the heart of the Bible. Okay. But if you see the proper kidney and its anterior end opens here, this opening is called as pericardial aperture. Where it is opening? It is opening in the fluid filled pericardial cavity. So this is the cavity, in this it opens. This is that opening renopericardial cavity which opens into the pericardium. Okay. Now this is another part, this upper or dorsal part is upper or dorsal arm, which is non-glandular, lined by the supra. Here is the location of the suprabranchial. Now here, actually in the section, see, this is the location of the supra. This is the location of the suprabranchial chamber. This is the location of suprabranchial chamber. Now this kidneys, that end, means this end, aperture is called as renal aperture. Renal aperture opens. हे बघा कुठं ओपन होते, lined by the suprabranchial chamber of the inner gill lamina by small renal aperture. म्हणजे where this is the inner gill lamina. suprabranchial chamber इथं पण आहे आणि इथं पण आहे. तर कुठल्या suprabranchial chamber मध्ये, जो चेंबर विच इज अबव द इनर गिल लॅमिना. कशाने ओपन होत आहे? बाय द रिनल अपर्चर, बाय द रिनल पोर और रिनल अपर्चर इट ओपन्स इनटू. सो द ब्लॅडर ऑफ बोथ द किडनीज इज इंटरकम्युनिकेट बाय द ओव्हल. म्हणजे बोथ द किडनीज का म्हणलेलं आहे? बिकॉज दे आर लाईंग क्लोज टू इच अदर. दे आर कनेक्टेड, हिअर दे आर कनेक्टेड बाय a small oval aperture. Okay. Now mechanism of excretion. How mechanism of excretion. Now ventral portion, that is kidney proper, it extracts main guanine and the other nitrogenous waste products from pericardial fluid, because its anterior end, that is renopericardial aperture, is in the pericardial fluid, as well as from the blood supplied to the kidneys. So from both these locations, the kidney proper is extracting the nitrogenous waste products. The wall of the pericardium are supposed to secrete waste material from the blood into coelomic cavity. The ciliated epithelium of the bladder produce outgoing current, thus carry the excretive fluid from the bladder to suprabranchial chamber, which leads to the excurrent siphon. So whatever is coming into the water, which is coming into the suprabranchial chamber, that is all the time it is thrown out, it is going out through the exhalent siphon. Mind it well. Suprabranchial chamber are communicated with the external environment through exhalent siphons. Now, this is one part, that one function that kidney is performing of the removal of the nitrogenous waste products. But another main function is osmoregulation. So osmoregulation is one of the important. And last time also I have asked you the question regarding what do you understand by the word osmoregulation. Yes? Can you answer me? Do you know the meaning of the word osmoregulation?
Osmoregulation means maintenance of osmotic pressure in outer and inner surface of body.
maintaining the osmotic pressure. Now this, why this osmotic pressure is generated, why it is created?
Presence of salts.
Presence of
Salts.
salts. Okay, so what
And
is the situation here in the freshwater animals?
in freshwater animals, osmotic pressure in their body is high.
Concentration of body fluids is high. Okay.
Is high.
Ah. Then the surrounding medium, isn't it?
Yes.
Yes. So what happens here? What difference do you observe here because of this difference in the concentration of the fluids?
Bivol is higher than its surrounding water medium. Concentration of body fluid in
Yeah.
bivol is higher than its water surrounding medium.
So how, what is the tendency of then water, where it is flowing, from
Uh,
body to surrounding medium or from surrounding medium to the body?
from body to surrounding medium.
No, exactly
Uh,
opposite you have told.
Flow higher.
Be- yes,
That is
because
to
of the, yes,
aquatic water medium to body of bivol. That
yes,
is
from
slow to
from water, surrounding water to the body of Bible.
bivol.
Okay. So here what the problem is to remove excess amount of water, isn't it?
Yes.
Yes. And that is done by the kidneys. So this excess amount of water, so maintenance of water and salt balance in the body is nothing but osmoregulation. Is that clear? So kidney also performs the function of osmoregulation. But this is the case with the freshwater organisms. And I have given you the example of the marine water animals also last time. Exactly opposite case happens with the marine water animals, isn't it? The concentration of salt is higher in the salt in the marine waters than the body fluids. So what will happen here?
The flow will be from the body of organism to its surrounding.
Its surrounding bodies. That means even though the animals are there in the water, they are facing the problem of water balance, isn't it?
Yes.
Yes, that is the problem in front of the marine animals. So they need to conserve the water in the body as well as the salts. That too, that function is also performed by the kidneys in the marine animal. Okay, I think you got the point. Okay,
Yes.
so excretion and osmoregulation is the function of this. Now, second organ is the Caber's organ. Now, this, see the location on the either side. So they are like situated in front of the pericardium. So to whatever triangular structure which are seen in the section, this in the section they appear to be triangular, but longitudinally they are running throughout the length of the body. This Caber's organ, again its name is given according to its discoverer. It appears to be reddish brown in color. Now these Caber's organ, they discharge waste material into the pericardial cavity. See, as they are in the vicinity of this pericardial cavity, they are releasing, what is pericardial cavity? It is a fluid filled cavity which is surrounding the heart. Okay. So now how whatever the discharge is there, waste material is there, which is collected by these Caber's organs, those materials are discharged in the pericardial cavity. From the pericardial cavity, as I have mentioned, at the pericardial cavity is filled and it is having a small aperture, where renopericardial aperture. So it is collected from this by this renopericardial aperture, brought into the kidney, and again from this kidney, from through this bladder, it is given through the renal aperture. From this where this renal aperture is opening, it is opening into the suprabranchial chamber, and from the suprabranchial chamber, again it is passed into the exterior through the exhalent siphon. In this way, the process, the mechanism of the excretion occurs. So, excretory system, that here we have finished up with the discussion of the excretory system. Now, let's go towards the reproductive system. Reproductive system. As sexes are separate, but there is no sexual dimorphism. No sexual dimorphism means externally we cannot make out whether the animal is male or female. Okay. So gonads, where these gonads are located? They are paired, located above the foot in the visceral mass amongst the coil of intestine. Now see this diagram. This is the foot, above the foot, in the visceral mark, among the coils of the intestines, in coils of intestine. Now, see, whatever the structures are there, here, round structures, these are nothing but the gonads. Now, during breeding season, they become enlarged and they are brightly colored. How we are going to recognize? By the color, the gonads appears whitish in male, while generally we call them as testes, and they are reddish in female. We call them as ovaries. In both, they are composed of branchial tubes lined by the coelomic epithelium. And this epithelium, which produce sperm and ova respectively, means testes produce sperms and ovaries produce ova. Gonads of each side has a short gonoduct which opens into suprabranchial chamber. Kutla again of inner gill lamina. If you observe this, आता here is the foot. Above the foot, here is the location of the gonads actually. Here we are seeing the lateral view. असंच दुसऱ्या बाजूला सुद्धा गोनॅड्सची लोकेशन आहे. On the both the sides, on the either side of the foot, gonads are located. याच्या इमिजिएट ह्याच्या विसिनिटी मध्ये कुठले गिल लॅमिन आहे? इनर गिल. त्याच्यावर आउटर गिल लॅमिन. While studying the mantle, we have come across these two structures. So whatever gonoducts are there, these gonoducts, they open into the suprabranchial chamber of the inner gill lamina, of its side, called as genital aperture. So situated in front of the in front of the renal aperture. Okay, so this is the location of the gonads. Now, fertilization. How the fertilization occurs? Spermatozoa are released into suprabranchial chamber, then passes into the cloaca, and finally escape with outgoing water current through exhalent siphon. Means here suprabranchial chamber, इथे exhalent siphon, इथून सगळे परत sperms are given out, they are thrown out. Some of these sperms, they enter. Now whenever these sperms are released in the water external, the female, if that female is in contact, is in vicinity of the male, from this external environment through the water enters कुठं जाणार आहेत the sperm enters the mental cavity of female with water current through inhalancy form. मग त्या फिमेलच्या इनहेलन्सी फॉर्ममध्ये खालच्या इनहेलन्सी फॉर्ममधून water enters here and comes into the mantle cavity and the inhalant siphon incoming water बरोबर पाण्यामध्ये असलेले sperms they enter into the female body. When it enters into the female body, ह्या इन्फ्रा इथं आल्यानंतर इथं लोकेशन आहे गिल्सची लोकेशन आहे. गिल्सची लोकेशन आहे म्हणजे लॅमिना आहे. लॅमिना कुठला आहे इनर गिल लॅमिना आहे. इनर गिल लॅमिना कसा आहे आपण वी हॅव स्टडीड द स्ट्रक्चर ऑफ इनर गिल लॅमिना. Inner gill, the surface of the inner gill lamina is showing the apertures. What these apertures are called as? Yes,
Ostia.
ostia, correct. So we have studied the structure of the inner gill lamina and I have compared the structure of inner gill lamina with that of the conductor's bag last time. Isn't it? This is having two pouch-like, this is pouch-like structure. Inner gill lamina and this is outer gill lamella, and both see these lamina they are closed at the anterior side, at the ventral side and at the posterior side, only opens at the dorsal side in the suprabranchial chamber, but this is divided into compartments. कशामुळे कंपार्टमेंट्स आहेत ह्या कंपार्टमेंट्स काय म्हणले त्याला दे आर इंटरलॅम्युलर जंक्शन्स आर देयर which divides the gill lamina vertically into compartments and these compartments are called as water tubes. Okay, now listen. Now the eggs shed by female in suprabranchial chamber are carried into water tubes. म्हणजे इथून हे कारण दोन्हीकडचे गोनाडक्ट्स कुठं ओपन होत आहेत दे आर ओपनिंग इन द सुप्राब्रँचियल चेंबर. पण गोनाडक्ट ऑफ मेल काय करतायत व्हॉट द गोनाडक्ट्स ऑफ मेल आर रिलीजिंग sperms where the sperms are going
into water current.
They are following the water current and they are thrown out of the body, isn't it, through exhalent siphon. But what happens with the female gonadoct releases ova, but this ova immediately they are released in the suprabranchial chamber and they are brought into mantle cavity. See, they are brought opens in the suprabranchial chamber are carried into water tubes of आता इथे बघा कुठला गिल लॅमिना आहे आउटर गिल लॅमिनी अनेक वचन दोन्ही साइडचे where the egg ovas are deposited, they are deposited in the outer gill lamini of the respective side. मग आता फिमेलच्या बॉडीमध्ये काय आलेले आहेत स्पर्म्स कुठून आलेले आहेत बाहेरून आलेले आहेत isn't it, they are coming from the outside, isn't it?
Yes sir.
Yes, they are coming from outside through the inhalant siphon, they are entering into the mantle cavity, that is infrabranchial chamber. From this infrabranchial chamber they are taken in the water tubes through ostia into the water tubes. आणि वॉटर ट्यूब्स मध्ये ऑलरेडी काय येऊन बसलेले आहेत
Sperms.
ova they are already there in water tubes, so sperms are entering through this ostia into the water tubes, isn't it?
Yes.
And they are what they are doing, they fertilize the ova. What is the ultimate result after fertilization? A
Zygote
zygote is
formation.
formed. And this zygote which is formed in the water tubes, this zygote is remains surrounded by the vitelline membrane, and uh, if you observe it, it is having a small aperture that is called as micropyle. So now here the embryonic development begins as soon as the fertilization takes place. So development of the zygote takes place in the water tubes of the female कुठला गिल लॅमिना तर outer gill lamina. Okay. Development. The development of zygote takes place in the water tube of the outer gill lamina. These water tubes acts as a brood pouch, or these are called as marsupia. Yeah, sorry. They are called as marsupia. Marsupium. Marsupium or brood pouch. There is a group of animals which are having marsupium in which the young ones are kept. Can you name that mammal?
Kangaroo.
Yes, very good. Kangaroo is having a marsupium, and we often see the young one which is developing, and even though the juvenile, it comes and goes into this marsupium. Likewise, that's why it is compared with the marsupium. So, or in other words, it is called as a brood pouch in which the development takes place. Now, this gives swollen apertures to the outer gill lamina. Because as the growth of the embryo is taking place, the outer lamina is appearing little bit swollen. The zygote, after undergoing the embryonic development, is transferred into the larva. Now this is very much important. This larva is called as Glochidium larva. It is a question of multiple choice question. The larval form of bivalve is called as dash. So it is that larva is called as Glochidium larva. Now how this larva appears? See, Glochidium larva. It is minute. Appears about zero point 0.1 to 0.0, zero point 0.1 to zero point 0.4 mm in width. The shell consists of two triangular and porous valves. See how it appears. These are the two valves, porous valves, united dorsally but free ventrally. So this is the dorsal side. They are united but they are free. So these are the free ends, free dorsal, ventrally. The ventral free end of each valve is produced into the hook with many spines. See this. Produced into the hook. So this is the limit of the ventral side, which is produced into the hook. Then this hook which is having these spines. See these spines. Spines are present here. The shell encloses the body with right and left mantle lobe. See, this is the mantle. Right and left mantle, which is shown here dotted. Mantle lobe. The mantle lobes are very small and their margin bears on each side 3 or 4 groups of brush-like sensory bristles. See, 3 or 4 such sensory bristles are present on the inner side of this mantle. How it appears. The valve clasped together by the action of single adductor muscle extending between two valves. See, एकच मसल आहे. Adult मध्ये किती असतात? 2 असतात. There are 2 muscles present in the adult, but here in the larval stage only single adductor muscle is present. And because of the contraction and relaxation of these muscles, these two ends they clasp. उघडझाप continuous त्याची चालू असते. The foot is not yet developed, but mid-ventral surface of the body has a glandular pouch which secretes long sticky thread called as byssus. म्हणजे इथं हे ग्लँड्युलर पाउच आहे. या ग्लँड्युलर पाउचमधून या मिड व्हेंट्रल रिजनमधून एक असा थ्रेड बाहेर पडतो. दिस थ्रेड इज कॉल्ड एज बायसेस. ग्लँड्युलर पाउच. इथं ऍक्च्युली फूडची डेव्हलपमेंट होते, पण फूड इथं डेव्हलप झालेलं अजून नाही. ग्लॉचिडियम nourishes on the mucus secreted by the gills. आता कुठे आहे मग, where this glochidium is present, where this development is going on? Yes,
Marsupial.
in the brood pouch, in the marsupial, or in the water tubes of the gills. So gills often they secrete mucus. So whatever mucus is secreted, that mucus is used as a food for the nourishment of the Glochidium larva. When mature, they expel out. वॉटर ट्यूब मधून कुठं जाणार, सरळ ब्रँचियल सुप्राब्रँचियल चेंबर मध्ये येणार आणि तिथून ते एक्सरेल सिफॉन मधून बाहेर पडतात. अँड दे सिंक ॲट द बॉटम ऑर स्कॅटर्ड बाय द वॉटर करंट्स. म्हणजे बाहेर जाणाऱ्या पाण्याबरोबर ते पाण्याबरोबर वाहून जातात, तरंगतात थोड्या वेळ आणि बॉटमला सेटल होतात. Now larvae do not move or feed. एका जागी ते स्थिर पडून राहतात. They can open and close their shell. फक्त उघडझाप ते करू शकतात. आता पहा. See, the entire structure as I have mentioned, two shells are there, united at the dorsal side. They are open at the ventral side. The margin of the wound is protrude into the hoop, which is having spines. The inner of this shell is lined by the mental lobes. Mental lobes at each inner side show the sensory bristles. Both of them are at the only one set of muscles is present, adductor muscles at the midventral side. In the midventral, this is the glandular pouch, which gives secretes the secretion, which is a thread-like secretion called as byssus thread. Now, further transfer to proper host. As I have told you that this Glochidium larva is present in the water, but it is sink down at the bottom, and only movement that is doing, that this larva is doing, is that it is opening and closing its shell or its valves. But now, to continue further existence, this larva must come in contact with the freshwater fish. आता life cycle, to complete the life cycle, it needs the intermediate host. And that intermediate host is fish. Otherwise, that larva will die. So those who come in contact with proper species of fish, they anchor its skin. आता जो thread जो आहे, तो byssus thread जो आहे, it can extend beyond the limit of the shell, and with the help of this, it can anchor, come in contact with the skin of the skin of the fish, fins or gills, and get attached. Then they are insisted by the overgrowth of the, मग जाऊन स्किनला चिकटतात आणि स्किनला चिकटल्यानंतर स्किनची वाढ होत असते, कंटिन्युअस त्याची त्वचेची वाढ होत असते आणि ती त्वचा त्या ग्लॉचिडियम लार्वावर चढते, ओव्हरग्रोथ होते. इट इज नाव लिडिंग द एक्टोपॅरासायटिक लाईफ. कसं? बाय रिसिव्हिंग द नरिशमेंट फ्रॉम द होस्ट फॉर 10 weeks. किती वेळाची अवस्था आहे ही, किती ड्युरेशनची अवस्था आहे? फॉर 10 weeks. इट इज लिव्हिंग द लार्वल फॉर्म in the body of the host, leads a ectoparasitic life in the about 10 weeks. During this period, it undergoes metamorphosis to develop into young bivalve. How this metamorphosis occurs? Metamorphosis. The parasitic larvae feeds on the growth by absorbing nourishment. त्यांना आता अन्न कुठे मिळतंय? होस्टच्या बॉडीमधून, होस्टच्या बॉडी फ्लुइडमधून, माशाच्या त्वचेच्या बॉडी फ्लुइड्समधून त्याला त्याचं अन्न. दे आर ऑल्सो नरिश्ड बाय डिसइंटिग्रेटेड लार्वल ऑर्गन्स. स्वतःचेच ऑर्गन्स काय होत असतात? आता डिसिंटिग्रेट होत असतात, कमी कमी होत असतात. त्याच्यातून ते नाश पावणारे जे ऑर्गन्स आहेत, व त्यात बायसेस थ्रेड असेल, ब्रिस्टल्स असतील, हुक्स असतील, ह्याचा सुद्धा अन्न पुरवठा म्हणून त्याचा वापर केला जातो. म्हणजे ड्युरिंग मेटामॉर्फोसिस कुठले स्ट्रक्चर्स जातात बघा. सेन्सरी ब्रिस्टल्स अँड बायसेस दे डिसअपियर. अ सिंगल ॲडक्टर बिकम्स नाव टू. आता आता जो आधी सिंगल होता तो आता दोन ॲडक्टर मसल्स डेव्हलप झालेले आहेत त्याच्यामध्ये. द मेंटल लोब आर रिप्लेस्ड बाय स्टोमोडियम. आता जे साईडचा जे इथं ग्रोथ, एंटेरॉन, स्टोमोडियम, इनव्हॅजिनेशन होतं इथं. कशाचं? रिप्लेस्ड बाय the mantle is replaced by the stomatia. Mouth is formed by the invalidation behind the position of the biceps and opens into. ह्या बायसेसच्या मागच्या बाजूला छिद्र पडतं तिथं. काय त्याला म्हणलं? स्टोमोडियम ऑर माउथ दॅट इनव्हॅजिनेट्स. आतमध्ये त्याची ग्रोथ होते, आणि आतमध्ये ग्रोथ होते म्हणजे एंटेरॉन. एंटेरॉन म्हणजे गट तयार होतो, त्याला गट सुरुवात होते. द एंटेरॉन रप्चर्स टू फॉर्म एनस. म्हणजे इथून तिथून एलिमेंटरी कॅनॉल तयार झाले. फूट मेक इट्स अपिअरन्स मिड व्हेंट्रली. आता इथं हे एंटेरॉन डेव्हलप झालेलं आहे, इथं माउथ ओपन झाले. आता हा जो भाग आहे सगळा हा फूटची डेव्हलपमेंट सुरू झालेली आहे. इथंच त्याच्या खालच्या बाजूला द माउथ अँड गिल्स अराइज फ्रॉम इट्स साइड अँड द रुडिमेंट्स ऑफ द विसल ऑर्गन्स ऑकर. म्हणजे आता हे हळूहळू बघा हे गिल्स हे यायला इथं सुरुवात ॲपिअरन्स होते. आफ्टर मेटामॉर्फोसिस द सिस्ट वॉल विकन. सिस्ट वॉल कशाची? की त्याच्या भोवती जे आवरण आहे, माशाच्या शरीरात त्वचेचं जे आवरण तयार झालेलं आहे, ते आवरण हळूहळू बारीक थिन होत जातं. अँड ब्रेक्स रिलीजिंग द इमॅच्युअर बायबॉल. सगळी ग्रोथ जी होती, लागणारे आवश्यक अवयव तयार झाल्यानंतर द सिस्ट वॉल स्किन रप्चर्स अँड इट थ्रोज आउट द बायबॉल, immature बायबॉल, and falls at the bottom. Now here they feed in the normal adult manner and gradually acquires the adult size. Like this. आता इथे एंटायर लाईफ सायकल तुम्हाला पाहायला मिळेल. कशी बघा. आता मेल आहे, मेल टेस्टीज आहेत मेलमध्ये. त्याच्या टेस्टीजच्या गोनोडक्ट्स आहेत, त्याच्या गोनोडक्ट्स मधून काय तयार होत आहेत? व्हॉट दिस गॅमेट्स, मेल गॅमेट्स आर कॉल्ड एज? येस.
Sperms.
Where these sperms are going?
They are going out of body through excellent siphon.
First they are released in the suprabranchial chamber, and from the suprabranchial chamber through the excellent siphon they are, see this, can you see these sperms? They are released in the water. Okay, now this is the female. फिमेल मध्ये काय होत आहे? इनकमिंग मधून वॉटर मधून काय स्पर्म्स जाणार आहेत? फिमेलच्या बॉडीमध्ये. फिमेलच्या बॉडीमध्ये जाणार आहेत म्हणजे कुठे जाणार आहेत? तर इन्फ्राब्रँचियल चेंबरमध्ये. इन्फ्राब्रँचियल चेंबरमध्ये कुठं जात आहेत ते? तिथून कुठं जात आहेत?
Water tubes.
येस. कुठून?
Ostia, through ostia.
हा, थ्रू ऑस्टिया. वॉटर ट्यूब्स कुणाच्या? इनर का आउटर?
Inner.
इनर का आउटर?
Inner.
Fertilization, we have come across this. काय म्हणलं इथं? The eggs shed by the female in the suprabranchial are carried to the water tubes of the outer gill lamina. लक्ष देत आहे? कुठं? आउटर गिल लॅमिना. ओके. हा, मग आता आउट इथं काय होणार आहे, वॉटर ट्यूब्स मध्ये काय होत आहे, ब्रूड पाऊच आहेत वॉटर ट्यूब्स. ब्रूड पाऊच मध्ये डेव्हलपमेंट होते. कशापर्यंत, कुठल्या अवस्थेपर्यंत डेव्हलपमेंट होते?
लार्वल स्टेज.
काय नाव आहे लार्वाचं?
ग्लॉचिडियम लार्वा.
ग्लॉचिडियम लार्वा. मग लार्वाची ग्रोथ पूर्ण झाल्यानंतर हे बघा, ह्या फिमेल मधून हे काय बाहेर सोडलेत, आउट गोईंग वॉटर मधून ग्लॉचिडिया आर डिस्चार्ज. These Glochidia are discharged. These Glochidia, and this is the structure of Glochidium, magnified, very microscopic they are, so they are released. आता हे काय, they come and sink at the bottom. See, these are the spots, but they are having the capacity to open and close. हे जे दोन व्हॉल्व्स आहेत ते सतत काय काय होतात, बीट होत असतात. बायसस थ्रेड मध्ये वळवळत असतो, हे उघडझाप करत असतात. आता unless and until they come in contact with the freshwater fish, suitable fish, their life cycle is not continued. So once the fish come in contact with this, immediately with the help of this byssus thread, they get attached to the body of the fish, either to the skin, to the fin, or to the gills. टच झाल्याबरोबर बघा, इथं सुरुवातीला नुसतं टच होतात आणि हे त्वचेमध्ये त्याच्यावर ओव्हर ग्रोथ होते. इथं थोडंसं झाले, त्याच्यापेक्षा जास्त इथं ओव्हर ग्रोथ स्किनमध्ये बुडाला येतो आणि इथं पूर्ण आतमध्ये त्वचेमध्ये ग्लॉचिडियम लार्वा आत सामावून घेतलेला आहे. सिस्ट त्याच्यावर तयार झाले, एक आवरण त्याच्यावर तयार झालेलं आहे. आणि पुढची डेव्हलपमेंट इथं काय होत असते, मेटामॉर्फोसिस. मेटामॉर्फोसिस मध्ये इथं काय, आता अन्न कुठून मिळतं त्याला, त्वचेतून. इथं अन्न कशातून मिळतं त्याला, इथं
Body parts which are getting dispersed.
no, in the body of bivalve I'm asking. What is the nourishment in the body of bivalve, in the brood pouch, in the water tubes?
Mucus of gills.
Very good, from the mucus of the gill. But here, during the metamorphic stage, during when it is leading the parasitic stage, it is getting nourishment from the body fluids of the host, that is fish, and metamorphosis takes place. As well as whatever parts which are disintegrating, which are now disappearing, that is the byssus thread, sensory hairs, bristles, they are utilized as a food during the metamorphosis. So single set of adductor muscle becomes two, byssus thread is incorporated, and then the development of the enteron takes place, gut takes place, slowly the foot develops, and on the either side here of the foot the development of the gills occurs, outer and inner gill lamina also. And after the full development of the larva, metamorphosis of the larva, the cyst now, as it grows, the cyst wall, that is skin of the fish, becomes weak. And once the skin becomes weak, it ruptures and this young bivalve is released out in the water. And here it leads a normal life, it leads and grows into the adult. Against adult becomes sexually mature, the gonad develops, again the gonads produce the gametes, male and female. So male and female, they do their job. In this way, the life cycle of the bivalve is completed. Okay, have you followed?
Yes, sir.
Yes. So highlights of the reproductive system. Reproductive system, even though the sexes are separate, there is no sexual dimorphism. There is no sexual dimorphism. Gonads are there, but where these gonads are located? Where
Around
the
the
yes, above the
around the
foot, in the coils of intestine, in the visceral mass. Okay. Gonads are lined by the epithelium, and this epithelium produce gametes, sperm in male and ova in female. where these two gametes primarily they are released. They are released in the suprabranchial
Branch.
chamber, and again through the outgoing water current they are, in case of sperms, they are
Released
released
through.
outgoing water. But what happens in the female? The ova are passed into, ova are passed into
Water tubes.
water tubes. Very good, water tubes. Then how they are fertilized?
When the sperm enters into the mantle cavity through inhalant siphon,
Correct.
and then it comes through ostia into water tubes,
Very good.
and then ova meets to sperm and the fertilization start, and
Yes.
zygote form.
Very good.
Then zygote is again uh, comes into
Embryonic.
larval
Embryonic
embryonic
development takes place.
development. Embryonic development. Zygote is formed into larva.
Uh,
The bivalve larva is called as Glochidium larva, and the
yes.
larva is then nourished into brood pouches that is called as marsupia, where
Mm-hmm.
young ones are kept. That is larva, Glochidium
Very,
larva, and
very
get
good.
nourished in gill mucus.
Yes, yes, yes. And then after completion of larval development,
They are excreted out of female body into water surface, water bottom.
yes. Then how the life cycle is continued further?
The larvae have to get a proper host, freshwater fish.
Mm-hmm.
Then only he can further complete its own life cycle.
Yes.
When Glochidium larva gets a proper host, it attaches to the skin or gills of the fish and
Yes.
get nourishment by the fluids or body fluids of fish and
Yeah.
its disintegrated body parts
Very good.
like spices thread.
Yes. Fine.
After a proper development of larva of 10 weeks, it gets discharged by the body of host and starts its own life cycle at the when it at bottom and it becomes adult and then he
Yes.
start further life cycle.
Yes. Yes. Very good. Very good. You have understood. I appreciate you, Nikita. Nice. Very good. And thus
Yes,
the
sir.
life cycle is completed. Okay.
Completed.
Yes. These are the references. Okay. Uh, any questions still? You, if you want to ask me any questions, you may ask.
No, sir.
I think you have understood thoroughly, right? Okay. So, shall we stop?
Yes, sir.
Okay. Have a nice time.
Good day, sir.
Good day.